So the final presentation will be by Marie-Pier Lalonde on apoptotic exosome-like vesicles and their specific sets of nucleic acid shaped endothelial function. So Marie-Pier joined Dr. Marie-Josée Hébert's lab at the CRCHUM in 2024 with a BSc in biology from McGill. She is now pursuing a master's in molecular biology at Université de Montréal. Her research evaluates pro-angiogenic RNAs and apoptotic exosomes and their effects on endothelial function. So Marie-Pier is one of CDTRP's Rising Star Awardees, and her award is courtesy of Simon Fraser University. So thank you, and we look forward to it.
Thank you. So hi, my name is Marie-Pier. I'm a master's student in Dr. Hébert's research lab at the CRCHUM. And today I'm going to present a project titled Apoptotic Exosome-like Vesicles and their specific set of nucleic acids shape endothelial function. I was told it was the big button, right? Oh, side button? Oh, yes. Okay. So to begin, kidney transplant procedures lead to some degree of ischemia-reperfusion injury. And if it's severe, this can result in acute kidney injury. And renal IRI also induces apoptosis of peritubular capillaries, and rarefaction of these capillaries leads to a loss of renal function and is a key determinant of progressive chronic kidney disease. Apoptotic exosomes, named apoexos, are also released during an episode of ischemia-reperfusion injury. Now, what are apoexos? These are extracellular vesicles whose release is dependent on the activation of caspase-3, the major effector of apoptosis. They're smaller in size, around 30 to 100 nanometers, than apoptotic bodies, which range from 50 to 5,000 nanometers. And they lack histones and HMGB1, a nuclear protein, both of which are markers of apoptotic bodies. Instead, they carry the functionally active 20S proteasome and the LG3 fragment of the proligin protein, a proteoglycan. And finally, they're highly immunogenic, and then they can trigger the production of anti-LG3 autoantibodies. A characteristic of particular interest of apoexos is their distinct mRNA profile. As you can see, there's a set of highly expressed protein coding RNAs here in yellow on the heat map that are found in apoexos, but not in apoptotic bodies or in cells that were in normal or serum-starved conditions. And the most enriched mRNA belongs to PCSK5, a serine protease from the proprotein convertase family. Additionally, apoexos can transfer this PCSK5 mRNA to recipient cells, and these cells will then express not only the PCSK5 mRNA, but also the PCSK5 protein. So first, extracellular vesicles are known to be key to cell-to-cell communication. And secondly, proprotein convertases, through their post-translational cleavage of precursor proteins, are known to play an important role in human homeostasis and in a wide array of pathophysiological states. And so, knowing this, we wondered, do apoexos and their cargoed PCSK5 mRNA play a role in the maintenance of vascular integrity? Our hypothesis was that apoexos do help maintain vascular integrity after endothelial damage via the transfer of PCSK5. And similarly, our objective was to determine the impact of apoexos and PCSK5 on endothelial properties such as cell survival, cell migration, and angiogenesis. So our first question was, do apoexos inhibit apoptosis? Cells were in presence of either vesicle-free media or apoexos for 24 hours. To induce apoptosis, we serum-starved the cells for 4 hours, and then we counted the number of apoptotic cells following a Hirsch and pyridium iodide stain. In bright blue in the images represents the condensed chromatin, which is a marker of apoptosis. And so there was a significant reduction in the percentage of apoptotic cells if these cells had been in presence of apoexos beforehand. And so this shows that apoexos do decrease cell death. Secondly, we wondered, do apoexos increase migration? Migration was assessed via a wound healing assay. The open area was measured at the time a scratch was performed and 6 hours later to calculate the percent wound closure. And there was indeed a significant increase in the percent wound closure for the cells that were treated with apoexos. And this means that apoexos did increase migration in this assay. Thirdly, we asked, do apoexos increase angiogenesis? Angiogenesis was measured with a tubule formation assay where cells were seated on an extracellular matrix like hydrogel. And shown are representative pictures of the tubule's formation 6 hours later, which shows capillary-like structures. And apoexos significantly increased the number of segments, but also the total length of the segments and the number of nodes, leading us to the conclusion that apoexos do increase angiogenesis in this assay. And so our in vitro results show that apoexos promote cell survival, increase migration, and increase angiogenesis. Next, we wanted to know if PCSK5 had the same positive impact seen on these cellular processes implicated in vascular repair, because this would give us an idea of the contribution of PCSK5 to the effect of apoexos. And so now cells were transfected with either a control or a PCSK5 expressing plasmid. And here apoptosis was measured with a fluorometric caspase 3 and 7 activity assay. And so first, we see that our serum starvation did induce apoptosis. As you can see, there's an increase in the caspase 3 activity between the normal and the serum starvation treatments. And however, the caspase 3 activity was significantly reduced when we transfected PCSK5 in the cells. And so this means that overexpressing PCSK5 does promote resistance to cell death. Next, migration was assessed with the wound healing assay. And note that the red lines in the images show the initial location of the scratch. And so as you can see, overexpressing PCSK5 does significantly increase the wound closure and thus the endothelial migration. And finally, PCSK5's pro-angiogenic abilities was assessed with the tubule formation assay. And similar to the apoexo experiments, there was a significant increase in the number of segments, but also the total length of the segments and the number of nodes. And so PCSK5 overexpression did increase angiogenesis as well. So to conclude our in vitro results, overexpression of PCSK5 promotes cell survival, increases migration, and increases angiogenesis, suggesting that PCSK5 does contribute to the effect of apoexos. Our next question was, is the pro-angiogenic potential of apoexos and PCSK5 also seen in vivo? To answer this question, we used a unilateral Heinlein ischemia model. Ideally, we would have used a renal model, but it can be difficult to look exclusively at angiogenesis. And in this model of severe persistent vascular injury, reperfusion depends entirely on the development of new blood vessels. And so Heinlein ischemia was induced via femoral arteriectomy, and then apoexos were injected intravenously at several time points before and after the surgery. And PCSK5 was injected intramuscularly on the day of the surgery only. And our hypothesis was that apoexo and PCSK5 injections would lead to increased blood flow recovery in the ischemic limbs. So here we have images of the mouse hindlimbs taken with the Doppler laser. In red and blue represent the highest and the lowest blood flow respectively. And vascular reperfusion was measured as a flow ratio of the ischemic limbs that we performed the arteriectomy on versus the non-ischemic limbs. And there was a significant increase in blood flow recovery of the ischemic limbs for the apoexo injected mice at 7, 14, and 21 days. We also measured capillary density, as the growth of new blood vessels is a key element of blood flow recovery, and immunohistochemistry staining of the ischemic muscle for CD34, a marker of endothelial cells, show that there was an increase in capillary density for the apoexo injected mice at 21 days. And so all in all, these results do indicate that apoexos favor angiogenesis in vivo. Now, again, we want to know if PCSK5 also shows this pro-angiogenic potential in vivo. And indeed, injecting PCSK5 in the mice after the arteriotomy led to a significant increase in the blood flow recovery at 7 days. So to summarize, we've shown that apoexos and PCSK5 help regulate microvascular repair pathways in vitro. And now we've also shown that the pro-angiogenic effect seen in vitro can be reproduced in vivo, as there is increased neovascularization after severe vascular injury when we inject the mice with apoexos or PCSK5. And so these results suggest that PCSK5, the predominant mRNA found in apoexos, may play a crucial role in facilitating the recovery of the microvasculature after severe vascular injury. The next step would be to validate these findings in a renal model, taking into consideration all other aspects of renal physiology that come into play. But ultimately, this work offers new insights into microvascular restoration mechanisms that could be of potential use for preventing microvascular rarefaction. And I'd like to finish by thanking Dr. Hébert's research team, as well as Sylvie Dussault from the lab of Dr. Alain Fifard.
Hi, this might be basic, but I was wondering why you choose to use HUVEC and hyaline as a model for the kidney instead of like using something that's more similar to kidney vasculature and endothelial cells, as well as like using like slices of kidney models instead. Thanks.
I think because our goal at this step was really just to look at neovascularization. And so we wanted a model that kind of excluded all other factors. Another model our lab uses a lot is just a kidney ischemia reperfusion model. But then we're looking at, for example, if we would have done the injections in that model, we would have looked at like how much we prevented capillary rarefaction and endothelial like function and damage and fibrosis. But here we really just wanted to look at angiogenesis and the growth of new blood vessels. So I guess that's why we used that in vivo model. And in terms of the cellular assays, why we use the HUVECs, mainly because they're endothelial cells. And so we're looking at like microvasculature health. So that's why we use the HUVECs.
Yeah, maybe I missed this. What's the substrates of PCSK5?
There's several substrates. Actually, I have a slide. There's like growth factors, and it also like has substrates involved in the extracellular matrix. And so are you asking because you're thinking about like maybe the molecular mechanisms that could explain the effects?
Inside the cell, right? A lot of these are angiogenic factors.
Exactly.
Yeah.
So, right. And we haven't yet like explored the specific, like experimentally, the molecular mechanisms that could explain what we've seen in our assays. But we've done like some proteomic analyses, hinting that it could be due to like cellular adhesion pathways, because those are upregulated in the PCSK5 overexpressing cells, but that's to be explored.
And have you been able to get apoexos from cells where you knock down PCSK5 instead of overexpress?
That's actually another thing that we are doing, because right now I've shown like in parallel the effect of apoexos and PCSK5 overexpression, but of course that doesn't specifically say that PCSK5 is crucial to the effect seen in apoexos. But so we did do some, so the way we isolate apoexos is we serum starve the cells, and then we condition the serum-free media with the cells, and then via ultracentrifugation we isolate the apoexos. But we also did isolate apoexos that came from PCSK5 depleted cells with an siRNA, and I don't have like the final results for those experiments, but we are looking into that.
Awesome. All right, I guess that's time. Yeah, thanks for the great presentation. I'd like to thank all the speakers.
Thank you.